Afternoon, everyone. My name is Scott Morrissey. I'm a PhD candidate from James Cook University in Australia. I'm working with Dean Jerry and Michael Kingsford on this work. So today I'm going to be talking about how I'm utilizing eDNA as a tool to better detect and study the ecology of deadly box jellyfish. Now, Chironex fleckeri, the Australian box jellyfish, is one of, if not the most venomous animals on the planet. It's responsible for hundreds of fatalities across its range. Its presence causes extensive beach closures, affecting tourism industries within Australia, and managing and mitigating the threat which it poses to human health and commercial enterprise is a wicked problem. However, despite this, considerable knowledge gaps on the ecology of the species and cubezoans in general exist. Now, one of these knowledge gaps is understanding on their population structures. Now, Kingsford, Schlafer, and myself wrote a review on this not too long ago and found that there's increasing evidence that some cubezoan species potentially have surprisingly small population stocks to the scale of bays and estuaries. However, from this review, we highlighted that we need to look into this further and actually look at where the jellyfish are physically dispersing to and incorporate aspects into this understanding such as cubezoan polyp locations. And speaking of polyp locations, this is another aspect surrounding cubezoans that we have limited knowledge on. Cubezoan polyps have only ever been found in their natural environment twice before, and that's for all 50-odd species. And understanding where they are is critical to furthering our understanding on their ecology, as polyps are the benthic source of medusae, so they give rise to the medusae, and so they play key roles in cubezoan population dynamics and population structures. Now, these knowledge gaps exist due to the challenges associated with actually detecting cubezoans in their natural environment. As we all know, they're quite elusive critters that are transparent and hard to find, and they are potentially dangerous and logistically challenging organisms to study. Additionally, the current tools for detecting and studying cubezoans have several limitations, which restrict the range of questions we can ask and investigate surrounding cubezoan ecology, specifically those knowledge gaps I just highlighted. Furthermore, these tools primarily focus on detecting the medusa stage of cubezoans with no effective means for detecting or studying the polyp stage in its natural habitat. And so to advance our knowledge and research capabilities, we need a new tool which can overcome the challenges of detecting cubezoans and address the limitations of the current detection tools. And luckily, environmental DNA, or eDNA for short, provides a potential solution to this. This technique allows you to detect animals in their natural environment by searching for their DNA signal rather than the animal itself. And so by simply taking a water sample, we can tell whether the jellyfish is present or not through just looking for its genetic signature in that water sample, instead of having to physically find and locate the jellyfish. So eDNA provides a tool that overcomes the challenges of detecting cubezoans and the limitations of current detection techniques by simply removing the need of having to actually physically find the jellyfish. And this is going to make studying their ecology, specifically those knowledge gaps I highlighted at the start of the talk, a whole lot easier. And so with eDNA, I wanted to see, can we use it to detect both medusa and polyp life history stages of Chironex fleckeri? Can I then compare the distributions of both life history stages based on eDNA to examine potential population stock boundaries of the species? And finally, again based on eDNA, can I determine the habitat which the species polyp stage is residing in? Now, to achieve this, I went to Port Musgrave, which is a semi-enclosed shallow bay located up the very top end of Queensland in Australia. And this area is known to harbour a considerable abundance of the species medusa stage during the jellyfish season. So it's a pretty great place to test out this tool. And additionally, this port is hypothesized to contain a population stock of Chironex. Now, this hypothesis is based upon biophysical models created by Jody Schlafer, which incorporated medusae swimming behavior and the movement of water to look at the distribution and movements of Chironex medusae throughout this area. Each of these dots you see on this map here represents a theoretical jellyfish, with the color referring to their abundance. And these models that Jody showed high retention of Chironex medusae within the port, with less than 2.5% of them being found to leave the area. And so this subsequently led to the area being hypothesized to represent a population stock of the species. To test this hypothesis, I undertook this sampling design here, where I sampled eDNA at spatial scales within the port along this gradient here, covering both beach and mangrove habitat, and I sampled at spatial scales outside the port, covering the beaches both north and south of the port's mouth, where these sites here and here were located 10 kilometers away from the port's mouth, so I could really see if Chironex medusae were moving out into these areas. And to ground truth any detections that I found, I undertook both SANET drags and or visual transects at each of the sites within the port to actually confirm that the jellyfish were there. So to ground truth the detections of the species that I find with eDNA. Now, looking at eDNA detections and quantities within the port during the jellyfish season, so I'm detecting medusae here, you can firstly see that we can use eDNA to detect the medusa stage of the species. And secondly, you can see that I found a high frequency of detection, with generally high eDNA quantities being found further within the port. If we look at medusa, which were physically captured or observed through the seine netting and visual transects I undertook, so medusae which were physically present, you can see that the jellyfish is quite spatially dispersed at sites close to the port's mouth, with 1 or 2 individuals being found. But down at this area called Red Beach, I found a hotspot of Chironex medusae. And if we look back at Jodie's model, you can see that the abundances that she found in her model correspond quite nicely with where I caught the highest number of physical medusae and found the highest quantities of eDNA. So both physical presence of the jellyfish and eDNA quantities agree with Jodie's model, that medusae are being largely retained within the port. Now, zooming out and looking at eDNA detections on the broader scale, you can again see that I found considerable detection within the port. I actually also found detection up both major river systems which flow into the port, so it's likely that the species is present throughout this entire area. But as I'm sure you've also noticed, I did find detection of the jellyfish outside of the port. However, this detection was of a lower frequency. These detections here certainly indicate medusae presence. I've got a backup laser pointer. These certainly indicate medusa presence, but just from this sampling, we can't tell where the medusae have come from. So to actually work out whether this port does contain a population stock of the species, we need to understand where the source locations of these medusae are. So where are their polyps? And conveniently, Chironex medusae are seasonal, and so they're only present in Australian waters during summer months, hence the Australian box jellyfish season. As a result, it provides an opportunity to try and locate polyps with eDNA, as any detection of the jellyfish during winter months when medusae are absent must be from polyps. So I went back to Port Musgrave in the middle of winter, undertook the same sampling design I showed you, and I undertook a very extensive amount of SANET drags to make sure that medusae weren't there, which they weren't. And as you can see, I was successful in detecting the polyp stage of Chironex using eDNA, which is really quite exciting. The last person who actually found cubozoan polyps spent 8 years physically searching for them, whereas with eDNA, we can quite efficiently work out where they're hiding just by taking a water sample. Now, here on the broad spatial scale, you can see that I found no detection of polyps outside of the port. So where I found detection of those medusae up here, that's not a source location of the jellyfish. The source location of the jellyfish for the area is within Port Musgrave. And if we look at this on the finest scale, you can see that detections were quite localized and that eDNA quantities were significantly lower than when medusae were present, which makes a lot of sense, as polyps are 1 to 2 millimeters in size, likely staying in the same spot, whereas medusae are up to 20 centimeters wide, swimming about, and we'd be pumping a whole lot eDNA out into the environment. So really quite confident that we're detecting polyps here. Sweet. And so with all of this evidence said, there's certainly support here for what was found with Jodie's biophysical models, that medusae are being largely retained within the port. And because of this and that polyps were found only within the port, there is evidence here to suggest that the area does represent a population stock of Chironex. And this adds to increasing evidence that some cubozoan species potentially have surprisingly small and isolated population stocks. Now, as eDNA can be utilized to detect the polyp stage of Chironex, we can for the first time look at the physical habitat and specific habitat type that the polyps are residing in. Traditionally, it was thought that cubozoan polyps resided within estuaries and estuarine conditions, but recent analyses on the elemental chemistry of cubozoan satellites and what we know on cubozoan physical tolerances has showed that suitable habitat for polyps may extend beyond estuaries to marine environments. And from where I detected polyps within Port Musgrave, you can see that I did detect them in both marine conditions and estuarine conditions, hence showing that they can and do occur in these two contrasting physical environments. Now, looking at specific habitats, what was interesting was that I didn't detect any polyps at sites characterized as mangrove habitat, which is really quite interesting. So perhaps mangroves aren't suitable habitat for Chironex polyps. Where I did detect polyps was at these sandy beach habitats, which definitely wasn't expected. But when I looked closer at these sites, I found that they contained expansive areas of carbonate reefs, often associated with oysters. And based on what we know about scyphozoan polyp preferences and due to the structural complexity of these carbonate oyster reefs, they likely make for suitable habitat where Chironex polyps can reside in crevices with appropriate water flow, bringing in food, removing waste, while also providing shelter from predators and sedimentation. Sweet. So, in conclusion, I found that eDNA can be utilized as an effective detection technique for Chironex fleckeri, and that it can be used to detect the elusive benthic polyp stage of Chironex with relative ease. And with this, we can look at the distributions of both polyp and medusa stages to infer population stock boundaries. Now, what does this all mean for the big picture? Well, we now have a powerful and effective tool to detect stinging medusae and to detect their elusive polyp stage and to identify source locations of deadly cubozoan species. And this is going to allow us to investigate the ecology of these animals in ways which we haven't been able to before, such as potentially being able to work out what triggers polyps to metamorphose into medusae, because we have no idea what causes that. And this is going to really help us, eDNA is going to really help us start filling in critical knowledge gaps surrounding cubozoan ecology. And the more that we know about their ecology, the easier it will be to tackle the wicked problem of dealing with the threat which they pose to both human health and commercial enterprise. So cheers.